Morning, everyone. So thanks for making it bright and early today. I will switch gears a little bit. So I will break away from the normal analysis workflow for spatial transcriptomics and talk about an emerging paradigm called cell segmentation-free analysis. So hopefully through this talk, I will explain to you what this means. We talked a lot about cell segmentation yesterday, and I'll give a few more examples, and then talk about how we can actually analyze this data without even thinking about cells and really trying to take gene expression in their spatial context. I have a few slides. I don't think we need them, but they're really nice slides of how we can represent transcriptomics at different levels. So over here, we have an anatomically accurate representation of the human brain with different cell types distributed spatially. So these will form tissues that have some sort of function which communicate between cells and allow the brain to function as it does. And traditionally, we've been doing mainly bulk sequencing. So if you're familiar with this kind of fruit smoothie analogy, this is essentially the fruit smoothie. And whilst it always does look like a mess, we shouldn't forget that actually most of what we know about molecular biology and gene expression comes from this type of data, right? The whole of Go, gene ontology, it's practically derived from stuff like this. And if you plan your experiments well, actually you can get meaningful data, right? Because you wouldn't analyze a whole brain, you might analyze a small section and try and find individual genes amongst those cells that you're profiling. Single cell was really taken off, so isolating these cells and getting individual transcriptomes after doing computational clustering on them. And now what we're kind of interested in is the spatial transcriptomics, right? So really getting gene expression in the context of the tissue, right? So I'm hopefully going to explain to you that seeing these individual Lego bricks doesn't actually matter. Knowing that it's red is more important than knowing it's a red brick. And the field is really, really rapidly emerging. I'm now at this point in spatial transcriptomics where I can't keep up with the literature because every 2 months there's a new fancy new method. And generally, the kind of movement in the field is moving towards having increase in size and resolution. So this is an image of a mouse embryo profiled by StereoSeq. And this is at half micron resolution, so 500 nanometers, practically full transcriptome. And these chips can get as large as 13 by 13 centimeters. This is massive. So if we think about technology such as Zenium, these can go up to 2 by 1 centimeter. Visium is in the order of just under 1 by 1 centimeter. So 13 by 13, we're talking about entire cross sections of human organs, which is typically not analyzed in the pathology context because they're used to constraining themselves to these slides. So the possibilities here are huge. We're also slowly realizing that we don't live in the world of The Simpsons and we actually are 3D. So actually taking these thin tissue sections only tells you about one plane of spatial biology. And now we're trying to do efforts of reconstructing pseudo 3D atlases or actually doing real 3D measurements. And yesterday we saw a very nice example of how certain technologies such as DAPI are also trying to implement multi-omics. So not just looking at the transcriptome, but also some other molecular readouts to allow you to phenotype and characterize your cells. But as a computational scientist, as a data analyst, essentially what you want to get to is this matrix at the end, right? For those that are doing single cell analysis, it's essentially the cell by gene. And now with spatial data, we get those X and Y coordinates. So we can really see where those genes are expressed in our favorite tissues. And in Laz's presentation yesterday, he said he nicely explained that there are generally 2 ways to get to this matrix of data, and one is profiling through sequencing-based methods, where you tag the mRNAs with regard to a spatial barcode, and then through sequencing you can recapitulate where these transcripts were identified. And one of the key parameters here is what is the size of the regions, these spots, these bins that allow you to resolve that spatial resolution. So the interspot distance is also very important, as is the size of these. And these typically range from supercellular to subcellular. And this makes a huge difference as to what the first computational tasks are once you get this data. The other approach is to use microscopy-based methods, so imaging-based spatial transcriptomics, where you can use in situ sequencing or in situ hybridization to identify precise locations of transcripts within your tissues. And then you get a map of all those transcripts inside your tissue. And through segmentation, so drawing the borders around cells, you can assign those transcripts to cells and start analyzing our cell by gene matrix, and doing some fancy downstream analysis on spatial pseudotime or whatever is new and hot in the field. And there are quite a few companies here that are offering this technology. So when we got into spatial, it was really homebrew techniques, and you needed access to someone that knew about this technology. But now, through companies such as VizGen, Merfish, Brooker Spatial Biology, getting access to these technologies is much, much easier. So I wanted to quickly just go over this concept of spatial resolution, right? So this also determines kind of how we go about analyzing your data. So I've just kind of distributed some cells across this fictitious piece of tissue on the screen. And for those of you that have kind of looked over the Museum of Spatial Transcriptomics by Leo Pachter's group, there are certain ways we can kind of think about capturing spatial biology here, right? So we can already see that there are certain structures, right? So you have the brownie cells here, this single layer membrane of pinky cells, and this mixed blue and gray region here. So if you want to do a very cost-effective methodology, knowing your tissue, you could actually do something where you do some sort of microdissection-based spatial transcriptomics. So you know these cells are roughly the same. So as this layer, you can use a laser or some other method to cut out these cells and profile them at the bulk level and analyze those. The most popular technology is Visium. And I like showing this slide because it really tells you what you're measuring, right? So this is really an underrepresentation of the density of cells that you would get. So you can really appreciate that in each one of these spots, the task of figuring out the gene expression is not trivial. It's not that you get mix of cells, you also get partial bits of cells as well. You can also get these methods that are in the range of 1 to 3 cells. Technologies such as slide-seq and slide tags, where you're roughly capturing a cell. But this also gives you this kind of awkward problem of inside one region, you could have a cell, or you could have 2 or even parts of other cells, right? So depending on how your spots are distributed over the tissue, over the capture region, could make this a potentially challenging task. Once you start getting to the subcellular methods, this is where it kind of gets slightly easier, because then you know each one of your bins corresponds to a cell part. You might have certain instances where in a bin, you have parts of 2 cells, but this is much less of a problem than what is observed in technology such as the original Visium. And then moving to imaging-based methods, you actually localize these transcripts. So you really find where they are inside of cells or outside of cells. So reviewing over like these different ways of profiling spatial transcriptomes at different resolutions, at different spatial resolutions, we can also think about what are the initial computational tasks. So if we have these low resolution methods, such as microdissection or Visium, which capture many cells, we might want to do some sort of deconvolution. But with these high resolution subcellular or single molecule based methods, you want to aggregate these signals into cells. And the most common way of doing this is essentially going through this procedure where you have your cells. Those cells will have transcripts, and through your imaging-based method, you will localize those transcript locations in your cells. But when you get your data, you don't see the cells, you see the transcripts. And then you use a computational method to figure out spatial patterns, or you use a counter stain to overlay onto your data. Here, I've used a very simple method of guessing which cell belongs to which transcript based on my best judgment. And you can roughly see some cellular architecture of tissue, which looks pretty reasonable when you compare this to the original tissue. So this step is really called, this is the part where you're actually doing the segmentation. So really drawing a boundary around a real biological feature that we hope is really representing the cell. And then we analyze, we calculate that gene expression, we annotate those cell types. But before we can do any analysis, the first major step here is really that segmentation. So if this segmentation is incorrect, your downstream analysis is incorrect. So it doesn't matter what methods you're using. And then comparing this to the original, there are some pretty obvious mistakes in my very low-tech manual segmentation. You can see in certain regions where cells are highly dense, if you have no staining of individual cells or no way to delineate the borders, it becomes computationally intractable to actually define those borders. So these can be arbitrarily large or arbitrarily small. Sometimes if cells have a weird shape, so they're not perfectly round, they can also become very tricky to segment. So like these yellow ones have distributed transcripts all over the place. And they're actually cells in the brain that kind of represent this type of feature, such as adipocytes. And if you misassign, you can also maybe incorrectly annotate your cell. So this has been annotated as a dark blue cell instead of a green one. So traditionally, cell segmentation works on computationally analyzing some sort of counter stain and using that to draw the border around the cell. So we talked a lot about DAPI staining, which is a staining that marks out the nucleus area, and then using a computational method to really draw around this. And the kind of reference algorithm in this field is water-shaped segmentation. This is considered like a classical computational approach. But over the last years, we've seen a rise of especially deep learning methodologies that can also perform this task. And we're also beginning to see some benchmarks that are coming through. But it's still fascinating to me that this very basic concept of drawing a border around a cell, so important for any downstream analysis, but we're only beginning to see serious reviews about this process in the last 2 or 3 years. So we've published spatial pseudotime before we figured out how to draw a circle around a cell. It blows my mind. And there are a couple of different reviews out there. I've just referenced one from Briefings in Bioinformatics. And one of the things I'm quite excited about are these general purpose segmentation algorithms. So segment anything is a Facebook segmentation algorithm that's not trained for microscopy data. It's trained for everything. And having these sort of foundation models and repurposing them seems like a really interesting approach. There is a segment anything for microscopy as well. But I think generally in the field, the one that seems to be generally performing extremely well, I can't see it here, Cellpose. So they had a couple of iterations of this method. I think we're now at Cellpose 3, and it also allows you to retrain the method based on your local data, and the performance here is generally top tier. So as I mentioned before, for a very long time, we've been staining the nucleus using DAPI. But this is not really representative of cell. It's somehow, I guess, a legacy thing that we did when we were just happy with finding the nucleus and assuming the cell was perfectly uniformly shaped around that nucleus and expanding it. But this really isn't the case. So we're kind of moving away from this idea of doing nucleus segmentation and starting looking at different types of markers of cells. Can you get something from the cytoplasm? Can you get something from the membrane regions and use that to actually more accurately delineate the borders of these cells? In a study with Mats Nielsen's group, one of the things we found was actually the approach that's typically taken in the original Zenium cell segmentation was pretty poor. So they would take this DAPI stain, they would use an algorithm to identify the nuclei, and then they would perform a uniform expansion to find the cell borders. So if you had a cell by itself, it would be practically a round cell. The expansion is pretty big. The default was 15 microns. It's really massive. And if you bump into another cell, then you just decide you have a perfectly straight line between them. So it has a lot of massive assumptions about where the nucleus is in relation to the cell, the shape of the cells, that cells are generally the same size unless they're close to each other, in which case they're constrained by their neighbors. And the general optimization parameter is we want to get as many transcripts inside cells. That's now the metric we want to optimize. So you are encouraged to make very large cells. And we compared this to a different algorithm called Beza, which does segmentation based on transcript location. So it doesn't explicitly need to use things such as counter stains, and it just looks at these patterns. And one of the things we found was taking the approaches such as Beza, which optimize the reproducibility of these transcript co-occurrences of the tissue, you get a very high score for what we call the negative marker purity. So how often are you observing markers of other cell types within your cells? but this comes at the cost of the proportion of transcripts assigned to cells. Whereas the Zenium approach manages to nearly assign every transcript to a cell, but you have a drop of marker purity. So you're now beginning to get contamination from cells that really should not be, contamination from transcripts which really should not be expressed in those cell types. Over the last year, the field has really taken a step further. So they've realized this DAPI plus expansion really doesn't work very well. And now they're offering different ways of staining cells. So here I've given the example of what Zenium offer, 10x Genomics Zenium, and they offer this concept of multimodal cell segmentation. So this is where they take multiple types of stains, including the DAPI nucleus, but also a cell interior stain and a boundary stain. And they have a tiered approach to segmentation. So if they can accurately determine the borders of cells, they will draw around those cells. This has the benefit of if you have a cell without a nucleus, you can still identify it. Or if you have a multinucleated cell, you can still identify it. And if you can't use the boundary stains, then it falls back to interior stains. So it uses this to delineate the absolute border of where the cells would be, and then it calculates the internucleus distance to delineate those. And failing that, if you can't use the boundary of the interior, then it will use the classic nucleus plus expansion. I think this kind of methodology is fairly new, and I don't think there are any robust evaluations of how well this works compared to other methodologies, but I would imagine there are people working on this. But it does really produce much richer, beautiful images of your tissue compared to just a simple DAPI stain. We also had a talk from a mention of this paper yesterday from Helena's talk, which is the idea of missegmentation and post-segmentation QC is now becoming a thing. Again, it's absolutely crazy that the first paper that really looked into this was this year, even though we've been analyzing this type of data for I don't know how long. But they now take mathematical approaches to determine whether you have contaminating transcripts within a cell and try to reassign those transcripts based on the cell specificity models. So we mentioned this a bit yesterday in terms of this concept of spatial doublets. So we talked about, I mean, in single cell biology, whenever you run your cells through a microfluidic system, you can accidentally get multiple cells inside one of your gems, and you have two cells contributing to the signal, which is commonly referred to as a doublet. For spatial biology, it's not really the case that it's a doublet, because a doublet really implies that there's two, whereas it kind of you can have a contamination here and here, right? So it's kind of spatial contaminations because your borders are not correct. I don't know if there's a better term that's being used in the field beyond spatial doublet. We try to call these XY doublets because, again, we're only looking in the X and Y direction. Whereas in reality, a tissue section is not 2D. It's 3D. Very, very thin 3D, right? So this is either 5 microns or 10 microns. But if we know an average cell is 10 to 15 microns and you have a 10 micron slice, it's pretty naive to assume that every cell is perfectly adjacent to every other cell at the exact same level. Practically impossible. So through a simple mathematical model of distributing 10 micron balls in a 10 micron slice, this is kind of what you see. I don't see many clear individual cells. So this concept of Z-type doublets of cells being on top of each other is a completely ignored problem until 2025, where we put up the bioRxiv paper, I think 1.5 weeks ago. So this is my last slide on cell segmentation. I actually didn't want to talk about cell segmentation. I want to talk about cell segmentation-free methods. So when we started going into this, analyzing our first data set, which was published from the group of Sten Linnaeus from Lars and colleagues, we started looking into the shapes of these cells that we were trying to segment. And this is an astrocyte. It's a very common cell type in the brain. I honestly don't want to segment that. I honestly don't want to get an algorithm develop an algorithm that can accurately segment that. This is going to be super challenging. You have the nucleus here in the middle. You have all of these processes extending. The astrocytes play a structural role, so they're not just isolated. They're supposed to be touching other cells. And I don't think you see it so well, but you have these tiny patches of blue, and these are the nuclei of other cells. So this is really not a fun cell type to work with if you want to draw an accurate border. We mentioned before, you can sometimes have issues with overlapping cells. Sometimes when you're performing your staining, it doesn't work perfectly, right? Like this is an image of DAPI staining, and you can see some parts of the image. You can clearly see the cells and the nuclei, whereas other parts, they're really tightly packed. The signal drops, but you still have to try and analyze this. And also sometimes like the hypothesis of having single nucleus in a cell just doesn't work. If you're working with certain tissues, you can have multinucleated cells. So actually the cell border may encompass many nuclei. Or if you're interested in red blood cells, they don't have nuclei, right? So if you don't consider that, you'll completely miss these in your system. So what we thought about doing was, do we actually need to draw the borders of cells using some sort of cell stain? And to kind of motivate you to show you that this is indeed possible, this is an image of transcripts that were identified, kind of grouped into 3 categories: inhibitory neurons, excitatory neurons, astrocyte neurons, astrocyte non-neurons. And I'm sure by now every one of you has done the by eye segmentation, right? Based on transcript density, you can really figure out where those cells are likely to be. So using this as a very simple proxy of transcript density is a proxy for cell locations is a very strong premise to actually try and take a cell segmentation-free approach. So not trying to draw a border around the cell, but try to look at local transcript patterns and see if you can analyze those within their spatial context. So the cell segmentation-free approaches are basically broken down into 2 types of concepts. One is kind of based on graph-based modeling, where you kind of have a graphical model of spatial relationships between transcripts. And others kind of take a density-based approach. So figuring out within particular regions, what is the gene expression density, and using this to kind of classify. And further downstream, you can actually take this data and feed it into some sort of classification algorithm, right? So these are essentially producing pseudocells. So they're really not modeling any sort of cell landmark, but they may recapitulate what a cell might look like and allow you to analyze it. And similarly here, you get gene expression patterns in this kind of lattice space where each pixel will have its own gene expression. And then you can classify those pixels and get a map of your gene expression patterns throughout your tissue. So one important thing is cell segmentation-free methods are still segmentation methods, just a different type of segmentation. So if we consider these people in this image as cells, classical cell segmentation is a type of instance segmentation where you try and separate out those counts of individuals throughout your image. Whereas cell segmentation-free methods can fall into this area of instance segmentation if you have a graph-based model, or if you have a transcript density-based model, it's essentially semantic segmentation. So you try and see where are there people in your image, and you classify them. So if these people were touching, you wouldn't be able to tell that they were 2 people or 1, you would just know that they're people. And there's quite a few methods that exist. I haven't updated this for a while, but essentially, there are a couple of graph-based models. The most popular one is Beza, which is based on the hidden mark random field. These kind of transcript density-based models that try and look within particular windows and classify gene expression there. These are beginning to emerge a little bit. I think the most popular in this field is the tool Picture. And we've also released a couple of our own, such as SAM-Light, and recently Science. And we also heard yesterday there are a couple of like augmented approaches where they will take maybe some sort of cell segmentation to initialize and then use local spatial gene expression to refine the transcript assignment. This is by far not exhaustive. So there's really a lot that's being published in the last 2 years with regards to these methods. I'm just going with quick pros and cons. So cell segmentation-free methods are typically extremely fast. They have much lower computational resource requirements. You're not limited to stains, right? So you don't have any prior about how to segment cells that are complicated. and you're actually analyzing what you're measuring, right? You're not analyzing the cell stains, but really the transcripts that you have measured. So if you have an imaging-based transcriptomics method, could be that you've left out a gene for a cell type on your panel. Knowing that that cell is there, but it doesn't have transcripts is no longer useful, so you just analyze what you have. But there are a number of cons. I think the biggest one is the conceptual issue of, but where are my cells, right? What can I do with this data if I have a map of transcripts over your tissue? In the early days of imaging-based spatial, I don't think this really mattered because you couldn't really do things like differential gene expression analysis because you had 30 markers for 30 cell types. There's no dynamic processes that are being measured. But now I think since these large plex panels are playing a major role in defining experiments, I think knowing how you can analyze differential gene expression beyond simple classification of cell types is also super useful. There's also not very much you can do with the downstream analysis. When you have the map, the number of tools you can use is also quite limited. We're trying to work on that and try to have these segmentation-free tools also available for analysis, but this is kind of work in progress right now. So just to give you a bit of an overview of the tool we made, it's called SAM. That's where the basic concept came from, and then later re-implemented with a lot of performance improvements as another tool called Science by Nicholas, who's sitting at the back. Again, it takes this concept of transcript density is meaningful in terms of identifying cell locations. And the general approach that we take is through image processing. We can apply methodologies such as smoothing to change these individual measurements of transcripts into densities and using these densities as proxies of cell probabilities. So you can see that a lot of the background transcripts are kind of regressed out and you get a stronger signal within cells. For those of you who are interested, the smoothing that I applied here was the one that was default built into PowerPoint. So just smoothed it, right? So this is like a very accessible methodology that you can apply. And just to show you kind of what's happening there again is like you have these individual transcripts that are measured and you change this into a sort of density and you can use this in a probabilistic fashion for classification tasks. So the algorithm basically has 3 steps. You smooth the gene expression, provide some sort of reference cell type signature, and then generate the cell type map. The way we smooth is by applying kernel density estimation with a particular kernel. So the kernel determines how the image or the transcripts are smoothed, and we use typically a Gaussian kernel. You can change that to other shapes, but we don't have a clear reason why you would want to. And from here, you can translate your discrete molecules into gene expression distributions over the tissue. An important parameter here is how much do you smooth, right? So if you under-smooth, you don't really represent the shape of the cell. And if you over-smooth, you might be leaking gene expression into adjacent cells. We somehow found a bandwidth of around 2 microns to be universally good if you have a pretty good experiment. But if you have a very low capture rate, you might want to increase your sigma and your smoothing to really smooth between transcripts. So you would apply this kernel density estimation to every gene, and then you would get these smooth gene expression of every gene, and then you'd stack them up into what we call a gene expression vector field. I think the easiest way of thinking of this is it's essentially not a cell by gene table, but a pixel by gene table after smoothing the gene expression. In the same way, in image classification, you have channels of RGB where the distribution over pixels is kind of continuous. We now have gene expression values in this kind of same plexity as the gene panel. And then you kind of have this highly multiplex vector field that you can now work with and start classifying. One of the steps we do to actually reduce computation is to define a threshold of gene expression. So if there's not enough expression within a particular position, you just cut it out and you don't try and classify it. So these will be lowly expressed regions in the tissue, probably not originating from cells, and it makes the computation faster. Then you need to start relating those patterns to known gene expression profiles. So if you have a single cell gene expression experiment that's done already and you know the expression profile of T cells, you can use that directly. or you can figure it out from the data itself. So the approach we take is through identifying local maxima of gene expression signals. What I mean by this is, let's imagine that this is a representation of 3 cells, and we have some transcripts. After we apply our kernel density estimation, we get these smooth gene expression patterns, and then we look for local maxima. And we assume if the signal is high in a local region, it's probably the center of the cell. And if we take the gene expression at that pixel, it's very likely, or we hope, that it's representative of the cell's gene expression pattern. So this is another cheap way of actually identifying positions that are representative of cells within your tissue without any sort of reference. We did this in the mouse brain, so actually the correlation between the cell types is quite nice, and we got a very nice t-SNE map. So if you don't know what t-SNE is, this is something that preceded UMaps for low-dimensional representations. And if you know what t-SNE is, then you know how long ago we did this. And then once you have your kind of gene expression matrix of cell types by genes, again, it could be from your single cell experiment or from your spatial experiment, you can start comparing your vector fields and using any sort of classification method. I mean, we don't use classification, we actually just use Pearson correlation, which seems to work very well. You can start classifying regions of your image of whether they belong to the orange cell type, the blue, or the green. Continue over all of your cell types, and you generate essentially a cell type map. So whilst these look like individual cells, they're not distinguishable because it's classified at the pixel level, and these have been annotated to a type. And as I mentioned before, we kind of use Pearson correlation because initially when we developed this algorithm, the relationship between transcript and cell type was practically 1-to-1. You do not need fancy machine learning for this. One thing we haven't evaluated extremely well right now is how these methods work when you have extremely high plex panels and your gene selection for clustering is based on SVGs or HVGs. It might not be as robust, but I think generally it seems to work pretty okay. So we applied this to the an awesome fish data set back in the day. We got some nice cell type signatures that represented what we expected from single cell experiments. So that you can see the correlation between cell type gene expression patterns were quite good. The distinguish gene expression patterns of the different cell types is quite nice. This is not typically seen in spatial transcriptomic experiments. Everything kind of clumps up and then you have these stars. So our approach of really identifying distinct patterns seems to work quite well. We compared our cell type map compared to the original. Generally, we have a very strong likeness. What we do like to point out, however, is like the density of what's annotated is much richer, and in particular in this region here, which is the ventricle. We can zoom in a little bit to figure out why it wasn't annotated so well in the original data. And if you look at the original counterstains, the DAPI signal and the poly A, if you compare it to what you have at the bottom, you can see there's a massive drop of signal, right? So if you're really trying to use these stains as a ground truth for where cells are, you miss them. But if you look at the marker genes that represent those cells, they're expressed pretty nicely. So since SAM analyzes gene expression before it analyzes cells, we're able to annotate those cell types pretty accurately, whilst they're missed in the original segmentation-based approach. So this was in a small section of brain with 35 transcripts. And at that time, it worked quite well. SAM will not work on a Zenium run. I think it fails after 5 days. So because of this, and with the emergence of these really larger data sets with regards to physical size and panel size, we developed an optimized version of this called Science, taking very similar principles from SAM, but just taking every shortcut, taking every optimization, re-implementing code in Rust to make it work faster and actually get it appropriate for these types of technologies. So a typical, I think we analyzed the Zenium mouse brain sample within 5 minutes or 10 minutes. So this is full classification and producing a map at 1 micron resolution. Doing the whole mouse embryo and full transcriptome takes a bit longer. But we compare what we observe to the original. And generally, again, we see that same likeness, but there are certain regions that are quite different. And over here, you can see in our analysis, we get a lot more cells in these kind of spatial regions here and here. This is the spleen, and all of these cells are red blood cells. This is the umbilical cord. So again, another kind of ground truth. You would expect there to be a lot of blood in the umbilical cord. And this is all missed in the original analysis because they performed DAPI segmentation, and erythrocytes, red blood cells, do not have a nucleus. So they had nothing to draw a border around. So there's tons of signal for red blood cells, which is thrown away because they went for segmenting cells and not just making use of the transcript information. As I conclude my talk, I want to also mention that there are a few things that you can do even if you don't have cells. We've shown that if you just do some spatial window analysis, you can identify recurrent signatures over large tissue areas and then maybe construct these spatial clusters or domains. So this is something we managed to produce with SAM. It's very similar to what's observed in the original publication of that tissue and expectation of cell type distribution over this region of the isocortex. We're also in the process of releasing an algorithm called cell sonar. So what this works is for an individual cell type, it tries to identify spatial relationships with other cell types. But it does it at a pixel level. So it finds a pixel and it will say, okay, this is an alpha cell pixel. Then it will perform some sort of sonar scan. So it will see, okay, what do I see 1 micron away, 5 micron away, 10 microns away? And from this, you can build up a curve. It's a bit messy, and I don't have the actual animation that was in the PowerPoint. But what you can see is if we assume that this is a standard enrichment analysis where the value of 1 means it's around background noise, you can see at certain distances, you see local enrichment. So what you can see is for this particular type of alpha cell, at the region of around 200 microns and 390 microns, you get another spatial enrichment. And those cells are actually located in these kind of red bodies. And if you look at them, you can practically make out small triangles. So the distribution of these cell types within the tissue themselves is kind of at a fixed distance. So we can still use these type of maps to identify spatial relationships that are quite apparent. One of the things I like about this is this was a method that was actually developed for cell segmentation-free analysis first back in 2021, and then re-implemented for cell segmentation-based analysis. So it was put into ScamPy after it was cell segmentation-free. Unfortunately, this is not going to work. It's a video, but it's now a PDF, so you have a before and after. When we generate these sonar curves, we can actually put them into a neural network to try and generate and simulate data. So actually at the beginning, we have just some random pixels distributed, and over a number of iterations, we can start creating synthetic tissue. So everything inside this middle bit is indistinguishable from real tissue, but it's completely in silico. So we're very excited about this in terms of like, you can do de novo tissue generation, maybe model how organoids will develop, maybe initialize a normal tissue and apply some learned parameters from diseased tissue to see how disease progress over time. So we're hoping that like taking some of the deep learning approaches for image analysis, we can really translate this back to these to the cell segmentation free based methods and really kind of go beyond the typical cell-based analysis. All right, end of my talk. So the quick summary is bad cell segmentation can really ruin your downstream analysis. And cell segmentation-free approaches are really emerging as a new concept that should be run in parallel to cell segmentation-based analyses. And I've just put like a little bit of a workflow of how you might want to go through this. You might want to run a tool like Overlappy, which was the bonus part of the presentation that I won't go through, but you're free to download and do that yourself. And then run through a parallel analysis between cell segmentation-free and cell segmentation with newly introduced quality control tools and comparing between the two. And when you're happy with your cell segmentation-based analysis based on what you observe in your cell segmentation-free, then you can proceed with downstream analysis. So we'll now run a tutorial where we'll go over application of the science tool that was developed by Nicholas, analyzing a Zenium dataset of the mouse brain. And you will basically learn how to analyze gene expression patterns, define minimal gene expression thresholds, and create a cell type map. Lots of people to acknowledge from the talk. In particular, for this one, I would like to highlight Jumping Park, who developed the original SAM algorithm, Sebastian Thiesmeyer, that did a lot of work in addition, and Nicholas, who's also developed the SAM algorithm.